Muscle stem cells remain associated with their endogenous niche and can be manipulated easily, for instance through siRNA transfection. This method makes it possible to analyze muscle stem cells in culture in a setting which resembles the in vivo situation more closely than standard 2D culture models by preserving the niche. Begin by cutting sterile Pasteur pipettes with a diamond pen. For each mouse, use one large bore pipette with an opening of about 0.3 centimeters and a length of about 10 to 12 centimeters, and a second glass pipette with a small opening of about 0.1 centimeter and a length of approximately 22 centimeters. Smoothen the edges of both pipettes by holding the pipette tips in the flame of a Bunsen burner for 5 to 10 seconds with gentle movement. Immediately before use, coat both pipettes with sterile horse serum by filling the whole pipette with 2 milliliters of horse serum for 5 minutes, then ejecting the horse serum and allowing the pipettes to dry for 5 minutes at room temperature. Spray all equipment and